In transformation, think of a magical thing that happens. When some bacteria die and start to disintegrate, pieces of their DNA can be taken up by competent bacteria. There's something about these bacteria that allows them to be able to literally take up pieces of DNA from the dead cells. So they're adding new genes to their genome. So, can I give you a scenario about a unicorn or an opera singer? No. No? Okay, so imagine this. I'm an old plow horse. I hate my legs. I'm not very cute either. No wonder I hate my legs. And one day I'm working in the field and I look over there and I see something. And what is that? Oh my God, it's a dead unicorn. She's beautiful. I always wanted to be a unicorn. Slurp up her DNA, piece of her DNA. All of a sudden I grow a horn and I become beautiful. So I picked up her gene for being a unicorn, not just a regular horse. And I turned into a unicorn. My opera singing one is, well, Pavarotti's dead now, but I always liked Pavarotti. So you're driving up 301, you see this terrible car wreck, and you know how everybody has to look and see what's going on. So you're looking and, oh my God, that's Pavarotti laying dead in the road. He was killed in that car accident. Always wanted to be an opera singer. So you go over there and you suck up his opera singing gene. Ah! Now, well, he was a baritone, wasn't he? So that's how bacteria can get new genes. Remember what a strain is, a genetic variation of a species. This is how new strains develop. New strains might develop from mutation, but that's pretty rare. More likely, new strains develop, and more quickly, strains develop by horizontal gene transfer mechanisms. The second one is transduction. We haven't spoken about viruses yet, but viruses that infect bacteria are called bacteriophages, phage for short. So in transduction, a phage does the transferring of the genetic material. The phage infects the first bacterial cell. That bacterial cell has to replicate the phage genetic material. And in the process, cuts in the wrong place and actually adds a piece of the bacteria's own DNA to the phage DNA. Then when the phage is repackaged as a new virion and leaves that first cell, and then goes and infects the second cell, that piece of DNA from the first cell is carried with it into the second cell. So here's my analogy for that one. Okay, so you meet this guy, you have a new boyfriend, and you move in really pretty quick. You're living with this guy, right? And after a little while, you know, you've gotten everything you could out of him, and he's pretty worthless at this point, and you're sick of living with him, so you pack your bags and you're going to leave. But when you pack your bags, you accidentally get a pair of his underwear in your suitcase. And you meet another guy and you move in with him. And so you unpack your suitcase and you're like, oh my God, here's a pair of underwear from my previous boyfriend. Do you want to wear it? So you pick up a piece of genetic material from the first cell that you infected because you're a virus and you're a parasite. And then when you move to the new bacterial cell's house, you give that cell the piece of DNA that you picked up from the first guy. Okay? Of course, school might not approve of me telling you that. Then there's conjugation. Remember conjugation pilus? The first cell has a plasmid. One of the genes on the plasmid is the gene for the pilus. First cell hooks up with a new cell. The one that has the pilus is F plus, like male. The one that lacks a plasmid is F minus. They hook up, The F plus cell makes a copy of its plasmid, transfers it into the F minus cell. She now has the pilus because the gene for the pilus is on the plasmid, so now she grows a pilus, and she can copy the plasmid and pass it to another bacterial cell. So here's a nice artist drawing, but I love this. Here's a picture of it actually happening. And notice, I think, these are two different types of bacteria. This one's longer, this one's a little chubbier, this one has a lot of flagella or pili. This is the conjugation pilus, these would be fimbriae, probably. And then this one I can't really tell, but it doesn't look like it has any flagella or fimbria. Okay, so third one is conjugation, which requires a pilus. Conjugation. Right. A magical transformation. You need competent cells to do the magic. Transduction, you need a phage to do the transferring. Conjugation, you need to have a pilus, and the gene for the pilus is on the plasmid. Okay. I think I took this question off your test, but I just got to show you this experiment anyway. Let's see transformation. Upstairs, I have a couple of Petri plates of bacteria that we did transformation experiments with. And what was transformed was a pigment gene. We got the bacteria to take up different genes for pigment. And so then, with the different strains, some are yellow, some are pink, some are purple, some are blue, they used the 4 different strains of transformed bacteria to draw pictures on their Petri plate. So then when the bacteria grew, it was always pretty colored. So I'll show you those upstairs. But this is the first experiment with transformation that really solidified what was happening. In this experiment, he used Streptococcus pneumoniae, and this is Dr. Griffith. Where does it say about Dr. Griffith? No? Doesn't. Okay, this is Dr. Griffith's experiment. I don't know where it says that. Anyway.
Griffith's experiment.
Oh, there you go. Here it is. Thank you. Okay, so in his experiment, he had Streptococcus pneumoniae with a capsule. A capsule makes it more pathogenic because the capsule helps it stick inside the host so it can colonize, grow into an infection, and harm the host. Remember also, capsules prevent phagocytosis. They hide the antigens on the surface of the bacteria, and they're harder for phagocytes to catch. So, the more pathogenic strain has the capsule when it's alive. But if he heat treats it and kills the cells, the mouse doesn't get sick from it. So, he gets another strain that does not have a capsule. It's not pathogenic because it doesn't have a capsule, so the mouse is okay. Then what he does is he mixes the dead cells that have the capsule with the live cells that don't have a capsule. The live cells take up the gene for the capsule from the dead cells by transformation. He injects it into the mouse gets sick and dies. Oh, what a bummer. Seems like a perfect waste of a mouse. But that's what's happening in transformation. And this experiment was the groundbreaking experiment to illustrate that transformation does in fact happen, and it's DNA that's actually being transferred. Okay, that one. Oh, this is transduction. See your little bacteriophage, right? Lands on the bacterial cell, injects its genetic material. The host cell replicates the phage's genetic material, but accidentally puts a piece of its own in there. And when the new phages are released, one of them has the host cell's genetic material in it instead of the viral genetic material, or in addition to the viral material, and then gives it to this new cell. So this new host cell has this new chunk of DNA. That's transformation. Okay. Everybody good? Horizontal gene transfer mechanisms. Transduction, transformation, conjugation are a separate event from binary fission. Binary fission, cell copies its nucleoid, unless mutation happens, they're identical copies into two daughter cells. Horizontal gene transfer mechanism does not happen when the cell is dividing. It happens at another time. It's a separate event. Alright, we don't want that. I think we are moving on to, the antibiotics chapter. Let's The antibiotics chapter is largely covered in lab. There's a lot of concepts in this chapter, but we're going to do the practical application in lab. Now, when you come in tomorrow, you need to bring with you a document that's posted in Canvas in the lab materials for the Curtin Bauer Antibiotic Sensitivity Lab, and it's called Antibiotic Chart for You to Use. So just print it out and bring it. You're going to have to write your results on there, okay? Okay. Let me just check my notes, what I want to do here. This antibiotic chapter is fabulous. It's a little bit hairy because there's a lot in it. So I'm going to try to condense it for you, if I can.